Click for IB Biology. Here we're looking at stem cells, the process of differentiation, and with some examples of bones and hair follicles as containing stem cell tissue. There's a blank PDF of this presentation down in the description where you'll also find 10 questions with answers so that you can track your understanding of the content. And so here we have an image which is the cell we're going to begin with, which is the fertilized egg cell with a complete set of chromosomes, a complete set of genes, and the alleles that have been inherited. So let's see how this begins to differentiate into the individual, as shown here as the fetus. So the egg cell that we began with is a consequence of the fertilization of the egg cell with the sperm cell. And this cell then begins to divide, and the early embryo, the cluster of cells which have been formed, we describe these cells as being totipotent. Now this means that any one of these cells has the total capacity to differentiate, to divide and specialize into an entire organism, in this case a human being in my images. And so we could take any one of these cells away and it would continue its own path of cell division and specialization and differentiation and create individuals. And of course this is the basis of identical twins, what we call monozygotic twins, where they are genetically identical. The ball of cells is actually split apart and each part differentiates fully to form individuals. Now the totipotent embryonic cells then go through a number of cell divisions to form this ball of cells, which is known as a blastula. And this blastula is going to implant into the uterus wall to complete the development towards the child. Now at this point we're going to use the phrase pluripotent. And what this means is that if we look at this diagram in this zone here, we've got two parts. We've got these outer cells here, and then we have the inner cell mass. Now any of these cells here have retained the ability to form any of the embryonic membranes, such as the amnion or the placenta itself. So above my lines here, any of these can form any of those tissues, and they all retain the capacity to do that. What they can't do is what the inner cell mass cells can do, and that is to form the actual embryo itself, the fetus and the individual. And so these cells are now described as being pluripotent, which means that they have gone down the pathway of differentiation and there's been a split so that they retain the ability to form many structures, but not others. So there's a degree of specialization that can't be reversed at this point. Now the pluripotent cells will go through considerable numbers of cell divisions and gene expression, differentiation and specialization into the different layers and structures and organs of the individual. And at this point we have moved now to what is known as multipotent cells. Now within a multipotent cell, for instance, if we take an example here of the pancreatic cells, within the pancreas there are cells which have specialized into the various parts of the pancreas, but there are still stem cells in there, and they are multipotent. Now those stem cells have the capacity to divide and specialize, differentiate into any kind of pancreatic cell. So they still have the ability to form pancreatic cells, but not other tissues. And then if we come down here perhaps to the cardiac muscle cells, within the heart cells there are undifferentiated stem cells, and they retain the ability to divide and to specialize into any structure within the heart, but they cannot become skin cells or nerve cells or skeletal bone cells. Still we have those cells. Now, the interesting aspect of this is that whilst we associate stem cells with embryos, within adults and mature individuals, we still retain stem cells within our organs, which retain the capacity to be multipotent and to replace damaged cells or cells which require a correction. And so we still have stem cells in the mature organism. How those stem cells are switched on and off is the focus of a great deal of medicine at the moment. If we take a look at some specialized examples of stem cells, which is called a stem cell niche, if we look at bone tissue, such as our large leg bone, the femur, the marrow contains two types of cells, HSC and MSC. So the hematopoietic stem cells, they have retained the capacity to divide either into red blood cells, leukocytes as in white blood cells, or indeed as to platelets. So these HSC cells can do either of these three cell types, as these are stem cells. The mesenchymal stem cells have retained the ability to form the cartilage tissue that we associate with the ends of bones and various other features. And interestingly, they also have the capacity to form adipose fat cells, which is a very interesting idea. So our femurs and large bones such as the pelvis retain this stem cell tissues, which is often a very important source of tissue when it comes to the treatment of diseases such as leukemia. And then let's take a look at another stem cell niche where we have hair follicles, which is a surprising location for stem cells. We have here in black is the hair structure itself, and down at the base of this structure with the dermal papilla where the hair will grow. Halfway down the follicle tube is a region called the bulge, the follicle bulge, and this contains our stem cells. Now, bulge stem cells have two different pathways of specialization. One route allows them to migrate down and into what is called the hair matrix, where they will cooperate in the dermal papilla for the actual growth of hair itself. So they can differentiate and specialize to hair growth. The second pathway is this one here, where the cells from the bulge can migrate up into the epidermal tissue, that is our skin tissue, and there they can divide and form the various epidermal skin cells, and this gives us part of our ability to heal wounds and would be seen when we're actually forming scar tissue when we have cut ourselves or damaged ourselves in some ways. A very, very interesting consequence of our hair follicles. Two examples of stem cell niches. There are many others. Here's the link to the next video in this topic of cell specialization, a link to the channel, and of course to the playlist.